Now, when I'm doing my study, I could blind people. And what blinding people means is, and I'll explain this using an example. Let's say I have a new drug and I want to see whether this drug is effective. I could do a single blind study. And what a single blind study is that so there's the examiner, which is me, the researcher, and then there's the subject. The subject are the participants, okay, the people that have come to my study that are going to take the drug. So single blind study means, so here we're saying single blind, subject blind. That means the subject is blind. And what that means is that the subject or the participant has no idea what drug they're getting. They could get the placebo drug, the fake drug, or they could get my new drug. So I know what they're getting. I know whether they're getting the fake drug, which is the placebo drug, or whether they're getting my actual drug to see whether they lose weight, for example. Let's say I'm interested in weight loss. I give them a drug. I know that they're given a placebo drug, for example. Let's say hypothetically they're given a placebo drug. I know that, but they don't. They think they're getting the real drug. So that's subject blind, where the subject has no idea what type of drug they're getting, whether it's placebo or their actual drug. Then we could have single blind, examiner blind. And this is where I, the researcher, have no idea what drug they're getting, but they themselves know. So he knows that he's getting a placebo drug, or he knows that he's getting the real drug. So can you see how when you have single blind study, there's a high chance of bias? Because if I know that this guy is getting the placebo drug, for example, I'm not going to pay much attention to that because I don't care about him so much. I care about the people that are getting my drug. So I might dismiss him. I may not look at his results carefully, and that's bad. Whereas this one, if I don't know, but the subject knows, so let's say the subject is taking a placebo and he knows it. Do you think he's actually going to keep taking that pill? He might lie to me and say, yeah, I took that pill, but he actually didn't. And because he didn't take it because it's a placebo pill, because he knows that whether he takes it or not, it's not going to make a difference for his weight. So that's actually not really an ideal type of study, right? So single blind, subject blind, single blind, examiner blind, not really ideal. Double blind. So double blind is where neither of us know. I, the researcher, do not know what type of drug he got. He, the subject, also does not know what type of drug he got. So no one knows. And that's the best type because there's no bias. I have no idea whether he's taking the placebo drug or the actual drug, and he has no idea. And so when we compile our results, we won't have any judgment. We won't have any bias in the back of our mind. And that's the best way. And then we have open label where both of us know which drug they're taking. Okay, so the best type is double blind. Endpoint, which is a term you may see in your paper when you're reading a research paper, a dental research paper. So endpoint is basically what happens at the end of the treatment. So if I do a study on whether Listerine is working or not, the endpoint would be that I want to see that there is no inflammation. The endpoint would be that I want to see a reduction of gingivitis. So endpoint is what happens at the end. Did the treatment work or did the treatment not work? Statistical design, we're actually going to do a whole unit on this later on. So for now, don't worry too much about it. The way to test for statistical design or the way to know whether the study actually worked is by p-value. And p stands for probability, which basically means it's a number that tells you what is the chance that the study worked or what is the chance that the study didn't work. And again, don't worry too much about this. I'm going to explain this later on in a different lecture. So after reading the methodology, we need to ask ourselves some questions. We need to ask ourselves, what is the number of study subjects? Were there only 10 people that participated, or were there 100 or 1,000 people? Because remember, ideally, most of the time, the more people that participate, the stronger the study. Then we look at what is the duration of treatment. Was the drug given for just a week? And then a conclusion was made after that week? Or was the drug given for a year or 2 years or 10 years? And typically, the longer the duration, so if a drug was given for 10 years and data was assessed for a 10-year period, then that's good. That means that's stronger than something that was just a drug that was just studied for a week or 1 month, right? We get more information from 10 years as opposed to a week or a month. So the longer the duration, the better. We want to make sure that when they divide people into groups, it's always randomized, because when we randomize, we limit bias. And we want to make sure that when we're doing a study design, it should be double blind, where I have no idea as a researcher, and when the participants or the subjects also have no idea. Now, sometimes it's not feasible to do so, and so sometimes we may have to resort to single blind, but ideally, double blind is the best way. So after the methodology, we see the results, and the results usually have graphs that explain the data. So when you're looking at the results, you want to make sure that you understand what the findings are, and sometimes we like to see visuals. This is an example of a visual that is highlighting the main findings. We also look at a discussion portion in the paper, and the discussion portion is actually the most interesting one, because in this section, what the researcher does is they're explaining how the results fit into the scientific knowledge. They're explaining the significance of the results, and this is what they always say in the discussion. They always explain the limitations. So if the sample size was low, so let's say there were only 10 people that participated, they'll say that. They'll say that next time around, we should have 100 people participate. And so they'll say that the 10 participant is a limitation, small sample size, right? So small sample size means that there was a small number of people that participated. And typically, we like to see a large number of people. They may also suggest topics for further research. So as they were doing their research, maybe more questions have come in their mind, and they'll say that next time around we should look at this topic, or next time around we should look at XX topic. So other topics may be suggested for next time, or for research for next time. When you read the discussion, you want to find out what the main findings were, and you also want to see that they state their limitations. Did they state that there was a low sample size? Another limitation could be that they didn't randomize, and that could be something that they said that next time maybe they would like to randomize the groups. We also have a conclusion section, and in the conclusion, they conclude all the findings. And when we read a conclusion section, we want to make sure that it answers the research question. So if my research question was, did this drug help with weight loss? My conclusion should say, yes, this drug helped with weight loss, or no, this drug did not help with weight loss. So I should answer the research question. Finally, we have the references, and the references is important because we want to make sure that the references are recent and that they're up to date. So if we see references that are 10 or 20 years old, and that's really old, we want to see references that are more current. The more current the reference is, the stronger the article. Now here, we're talking about professional application, which means why is it important to us as hygienists? Well, the reason why it's important is because science is evolving every day. Think about it. Right now, we're looking at COVID, right? COVID-19. That's all over the news. Who would have thought that 5 years ago something like COVID-19 would affect us so much? So when we're researching about COVID-19 and how it affects dental treatment and how it affects clients and us as professionals, how we can get it, we shouldn't just be looking at the news. We shouldn't just be looking at the magazines. We need to be reading credible papers. We need to be reading credible journal articles. So when you find a paper and you read it, you may come across some conflicting information where one paper states something and this paper states something else. So use the skills that you have learned in this PowerPoint to make your own conclusions. Look at randomization, look at sample size, look at references? Is it current? Is it not? Look at the people that wrote that paper. Are they health professionals? Do they know what they're talking about? So use the skills that you have learned to draw your own conclusions. When you're working in a dental office, you will see dental reps coming in, and they will try to sell you products. When you're reading journals, you will see commercials, you will see ads rather in the journals that are trying to sell you product. What's important is that when you see those ads or when you see those dental reps trying to sell you a product, always, always, always ask for literature, ask for data to support the product, to support the product efficacy and safety. When you recommend something to your client, you want to make sure that there have been research done in the past to show that it works, and that the research that has been done is credible. And so some ways you can find out whether it's credible or not is that when you're looking at the paper, you want to make sure that the subjects have been randomized. You want to make sure that the study is recent, it's not from 10 years ago. Right, you want to make sure that the people that did the study are dentists or hygienists or health professionals who know what they're doing. So there's many things you should look for. So to conclude, anytime we as a dental professional are giving advice to our clients, we want to make sure that the advice we give are evidence-based advice or evidence-based decision, that they are in the information that we give them are the best type of care and recommendations. And how do we know that they're the best type of care and recommendations? Because we did our research. We did our thorough research. We didn't just go to Google and find information, because we know that Google is not an accurate source to get information. We actually went through the trouble of reading journal articles, and based on the readings, we then make a recommendation for our clients. So it's important that we know how to critically read, how to critically evaluate clinical research papers, because when we know how to do that, we can give the best possible care to our client.